Now, let's see about cell junctions. Cell junctions are the various components through which cells are linked with each other or with the extracellular matrix.
Cells don't like to live in isolation. Instead, the cells come close together, they interact with each other, and they create tissue. In this particular example, a layer of epithelial tissue, a skin tissue. So the cells kind of interact with each other and they stick to each other. And the way they stick to each other is by several cellular junctions. And these cell junctions could be of different types. So let's just quickly look at what are the broad category of cell junctions present between the two cells and what are the mode of communication between the cells.
So these cell junctions are categorized into three types based upon their functions. They are anchoring junctions, communicating junctions or gap junctions, tight junctions or occluding junctions. Anchoring junctions, communicating junctions and tight junctions are the three different types of junctions which are categorized based upon their function.
So one of the important junction is gap junction. It is also known as channel forming junction. So it is also a mode of transport between ions and other messenger molecules between the two cells. Now, adherence junctions are mostly structural support. They are providing the cells to stick to each other, and on the other hand side, these adherence junctions are anchored to the actin cytoskeleton such that two cells kind of mechanically intertwine with each other nicely. Now there are tight junctions which works like a sieve. So it closes the cell in such a way that even small molecules cannot pass.
Now let's see them in detail. Anchoring junctions. As the name itself suggests that these junctions anchor the cells. Anchor the cells in the sense they give a grip or support to the cells. And this anchoring junctions can occur either between two adjacent cells or between the cell and the extracellular matrix. And these anchoring junctions are abundant in those regions where the tissues occur constant wear and tear, such as the skin and heart. The anchoring junctions consist of two main components. They are cytoskeletal proteins and transmembrane proteins. And these act as an cytoskeletal anchor and transmembrane linker. Based upon the type of cytoskeletal anchor and transmembrane linker, which both are proteins, these anchoring junctions can be categorized into three different ways. They are desmosomes, hemidesmosomes, and adherence junctions. Desmosomes rivet the plasma membranes of two cells which are adjacent to each other. In desmosomes, the transmembrane proteins are called as adherents, whereas the intracellular proteins or the cytoskeletal anchor are called as intermediate filaments. Intermediate filaments can be either keratin or desmin. The cytoskeletal anchors, which can also be called as cytoskeletal proteins or intracellular proteins, they form a plaque at the plasma membrane, and the transmembrane links, they cross through the plasma membrane and they form a link with the transmembrane protein of other cells. In this way, the transmembrane links of two cells, that is, the adherents of two cells, link together to form this junction, making the cells in a tissue tightly bound or attached to each other without getting dispersed in the extracellular fluid. The other type of anchoring junctions are hemidesmosomes. In hemidesmosomes, the cytoskeletal proteins and the transmembrane links, they are involved in forming a junction or a link between the cells and the extracellular matrix, such as basal laminae of the epithelium. The intracellular proteins or the cytoskeletal anchor is once again the intermediate filament, which is either keratin or desmin, which forms a plate, and the transcellular anchors here are called as integrins. These integrins, they bind with the extracellular matrix and give adherence of the cells to that of extracellular matrix. So, in this way, the epithelial cells are attached to the basal lamina. The other type of anchoring junctions are adherence junctions, and these adherence junctions have both cadherins and integrins as their transmembrane links, whereas the cytoskeletal anchors are actin filaments. Once again, the cadherins occur in anchoring junctions when a cell-cell binding occurs, whereas the integrins are found when the cells are to be bound with an extracellular matrix. And this sort of anchoring junctions will give the characteristic folding and bending nature of an epithelial membrane. The other type of
The next kind of junction we'd be talking about is adherence junction. As the name suggests, it helps the cell to adhere to each other, so that's the name, adherence junction. So adherence junction are actually formed of specific anchoring molecules known as cadherins. So here you can see in green there is cadherins. So cadherins have one extracellular domain which is composed of 5 subunits. It's projected outside of the cell, whereas the cytoplasmic domain is attached to the actin cytoskeleton. So as if it is anchoring the nearby cell to its cytoskeleton. So they are like deeply rooted. The connection is even strengthened in terms of the actin cytoskeleton. Now these particular cadherins are known to perform homophilic interaction, not heterophilic interactions. So cadherins from one side and cadherins from other side interacts with each other, and their interaction is basically calcium dependent, and cadherin attach with the actin filament via specific adapters like beta catenin, alpha catenin, etc. Now, so catenins are so important. So cadherins switch between an active and inactive state depending upon the concentration of calcium. When there is enough amount of calcium, cadherin gets activated and it can attach to the nearby cells and form this adherence junction. So cadherin is calcium dependent lectins, and they are super important for the adherence function, and or it kind of work like a velcro for attachment of two cells together.
Cell junctions are gap junctions. These gap junctions are also called as communicating junctions because they help in communicating between the cells. So here the cells can communicate with each other by transferring substances across them through these gap junctions, and these substances can cross the cell membrane of one cell and can enter into the other cell without getting in touch with that of extracellular fluid. There are some proteins called as connexins in the cell membrane, and these connexins unite to form connexon. The connexon proteins extend throughout the cell membrane and they form a gap junction when they come in contact with the connexons of the other cell membranes. And these gap junctions, which are formed by fusion of two connexons, they have a pore within them, and this pore has a varied size and polarity. Thus, making it specific for the transport of certain substances across the cell membrane. And these gap junctions are very essential in the body and play a very vital role. Example, contraction of, uniform contraction of heart muscle and signal transfers within the brain.
Now we'll talk about gap junction. Gap junction are like sieves, so we would like to tell them or think them as molecular sieves, and the smallest subunits that are making the gap junctions are known as connexin. These connexin heteromerize to form connexons. Now these connexons are channel made up of connexins. So 6 connexon uh, actually make these connexon channel, and from the top view you can understand this connexon channel can open or close. Now this connexon channel, while open, can allow the passage of several molecules and water from one cell to another cell, so they are kind of important in terms of communication. Now let's just look at some of their particular functions. So in kidney or any other cells, these gap junctions are very important because they help to filter out stuff. Now, in terms of synapses, some synapses, especially in invertebrate, synapses are gap junction coupled or electric synapses. Now, electric synapses, unlike chemical synapses, are very fast. Yeah, they don't have that degree of modulability or gain control, but still the electrical synapses are faster, are very important for fast nerve impulse transmission.
The other type of junctions are tight junctions. These tight junctions are also called as occluding junctions because they are mostly found in epithelia, and these junctions, they form a barrier by forming a close contact between the cells by binding the plasma membranes of adjacent cells, and this will restrict the movement of any water or solute across these membranes. And these tight junctions are also called as paracellular junctions. And these tight junctions are also called as paracellular barriers, which does not have any directional discrimination in the movement of substances across these cells. So the substances or the solvents can move from any direction through these occluding junctions and is mainly dependent on size and charge. Tight junctions are mostly seen in epithelia and are selective for solutes of differing size, charge and polarity, and there are various proteins which are involved in formation of these tight junctions, which can be broadly classified into either scaffolding proteins, signaling proteins, regulation proteins and transmembrane proteins.
First, let's talk about the tight junction. Now, the tight junction seal adjacent epithelial cells in a narrow band, and they seal it so tightly that even water molecules cannot pass through that. So if you zoom into this section, you can see the tight junctions are consist of these blue colored subunits which kind of stitch the membrane, as it appears that it is like stitching the membrane of the two cells. Now these blue colored subunits are actually claudins. So claudins are the subunit of tight junctions. Claudin or claudin, both are these tight junction subunit. Now, chlorine is a small 20 to 27 kilodalton transmembrane protein. They have 4 transmembrane domains, and they are found in many organisms ranging from nematodes to human beings as well, but they are kind of conserved in terms of their structure but not in terms of their generic sequences. Now let's just talk about the functionality of the tight junctions. So tight junction ensures that even water molecule or small ions cannot pass through the one side of the epithelial layer to the other side of the epithelial layer, and this also ensures that whatever passage of water molecule or ions etc would happen, it would happen via active transport or facilitated diffusion, not directly. So as if the tight junctions prevent the cells from leaking. For example, your hand has epithelial layer, which is your skin, and from on the top of the layer you pour some water, and what happens, like the water can go and dribble off your skin. It does not go inside of your skin and get mixed up with the internal tissue, right? So that is how tight junction is also protective for cell.
Now let's see about cell signaling. That is, how one cell can regulate the function of other cell by sending any signal. In human body, there are trillions of cells, and these cells can send and receive chemical messages to regulate the function of those cells which are either adjacent to them or which are very distant to them. Cells can receive this message, it can transfer the same information across the plasma membrane, and it can alter the functions within the cell. And before going into signaling, let's see about various basic concepts which we have studied previously. A signal molecule is nothing but a ligand and which acts on a receptor which is present on the same cell or on the adjacent cell, and this signal molecule will stimulate the receptor and activates the receptor, which produces a response. In this way, the signal molecules will produce the action on them or on the adjacent cells or on the cells which are distantly located. And this mechanism of sending or receiving signals can be categorized into 4 different methods. They are paracrine signaling, endocrine signaling, autocrine signaling, and direct signaling. Now let's see about them in detail. Paracrine signaling occurs in those cells which are very close to each other, and this transmission of signals or chemical messengers occurs through diffusion, and this sort of signaling will send messages where they can produce the response in a very short time, that is, which is very quick and exists for a very short duration. Example is synapse of nerve cells. Two neurons never attach directly. They come in close proximity with each other, and this proximity is called as synapse, and this transmission of signal from one neuron to another neuron occurs through paracrine signaling.
Paracrine signaling. The blue cell can release growth factors, for example, and all the surrounded cells here with the expression of specific receptors can now bind this ligand. This can then activate intracellular signaling, for example some downstream effects like gene transcription activation. However, cells in the near surrounding with no expression of this receptor, they cannot be targeted for communication. It is important to mention here that for paracrine signaling, the cells have to be in rather close range. The signaling molecules released by the donor cell here, they can't travel over a long distance. An example for paracrine signaling might be the expression of VEGF, so the vascular endothelial growth factor. They are secreted, and the surrounding cells with the specific receptors can then initiate angiogenesis.
Paracrine Paracrine signaling allows a cell to target another cell by a signal molecule that may diffuse between them. These cells are still close but need not be connected. The ligands in paracrine signaling tend to be rapidly absorbed and rapidly degraded. The ligands are typically not traveling far, as this is local signaling.
Synaptic transmission can also be considered as an example of paracrine signaling.
Synaptic signaling. In this case, we are talking about neurons and neurotransmitters. The upper part here is the terminal axon. It's also called the presynaptic neuron. The part on the other side is called the postsynaptic neuron, and in between we have the synaptic cleft. Our presynaptic neuron has neurotransmitters. So these neurotransmitters here are released into the synapse. So in this case, the neurotransmitter can bind to the receptors expressed on the postsynaptic neuron, and this binding of the neurotransmitters can open and close channels. Open channels can lead to ion flows, such as the rapid uptake of calcium.
The other common type of signaling is endocrine signaling. So in contrast to the paracrine signaling, where the transmission of signals occurs between two cells which are in close proximity, this endocrine signaling occurs between the cells which are far apart. And this signaling, it is mediated through messengers which are called as hormones, and the cells which produce these messengers are called as endocrine glands. Endocrine glands secrete these hormones, and these hormones travel slowly through the bloodstream and reach the target glands to produce their effects. And the response can be categorized as slow but long lasting, whereas the response in paracrine signaling is quick but of short duration, because the ligands which are involved in processing the information will be degraded there in paracrine signaling. And the other difference between paracrine and endocrine signaling is the concentration of the signal is high in the case of paracrine signaling, because the messenger which is released from one cell is in close proximity to the other cell, whereas the concentration of the signal in endocrine signaling is very low, because as this signal passes through the bloodstream, it becomes diluted and its concentration will be reduced.
When cells have to communicate but are in very large distance, so paracrine signaling is not possible because of limited diffusion. Let's make it more specific. We take different organs. The brain, for example, can secrete a hormone, but the target site, so the receptors, are located in the liver tissue. So these are two organs where we have quite some range in between, right? In this case, there's one type of signaling which is called endocrine signaling, where the donor cell will now secrete their signaling molecules into the circulatory system, and they will travel in the bloodstream. They can diffuse through the membranes once they reach their specific target cell to trigger intracellular signaling pathways. The secretion of hormones is a very good example here. Hormone synthesis takes place in glands. One example might be insulin. Insulin is produced in the pancreas and from there secreted into the bloodstream. The endocrine system helps insulin to travel over a long distance and reach muscle cells, for example, where insulin can bind to insulin receptors. That supports the uptake of sugars in the tissue.
Synaptic signaling, which specifically involves neurotransmitters in a synapse, is another example of this local signaling. Endocrine signaling can allow a cell to communicate with a target cell from far away. Signals may be carried in the bloodstream. Hormones released by certain types of endocrine cells are a great example.
The other type of signaling is autocrine signaling. As the name itself suggests, autocrine means self. So here, this signal which is released by a cell will act on the receptor or the which is present on the same cell, and this once again binds with that thing and it influences the activity of the same cell. Or this released signal will act on adjacent cells of same kind to produce the effects. Autocrine signaling occurs mostly during the early stages of development of an organism, where these cells will direct the adjacent cells to develop into their own type and perform their own function. Autocrine signaling also regulates pain responses and inflammatory responses. Autocrine signaling can also regulate pain sensations and inflammatory responses. Further, if any cell is infected with any virus, the signals from that particular cell will direct the cell to undergo self-destruction or programmed death, which is called as apoptosis.
Another signaling type here is the autocrine signaling. So in this case here, the cell can secrete a messenger, which can be a hormone, for example, and this one can bind to its own receptors on the surface. In that case, the cell has expressed the fitting receptor for its own secreted signaling molecule. We can find many good examples for autocrine signaling when we have a look into tumor cells. Most tumor cells show to be autonomous in terms of cell proliferation. Many of them show to develop independency of their microenvironment. This is why autocrine signaling is found a lot in tumor cells.
We also want to point out that a cell could just signal itself. For example, in autocrine signaling, a cell could secrete a certain type of molecule which then binds to its own receptor and causes a response. A cell releasing its own growth factor could be an example of this.
And the other type of signaling is direct signaling, which is also called as signaling between the cells directly through gap junctions. And here the transmission of signals will occur between the cells through the connexon proteins pores. We have seen the connexon proteins, which form a junction between two adjacent cells, have pores and allow the movement of selective substances which are based upon the size, charge and polarity. So in this way, substances such as ions move across the cell membranes to regulate one another. This transfer of signals between the cells will let the cells know what is the state of their neighboring cells, and it will accommodate to their condition.
Another form of cell communication is juxtacrine signaling. In this case, it's all dependent on contact. So in this case, we don't have the secretion of a signaling molecule. Instead, we have a membrane-bound signaling molecule on our donor cell. The juxtacrine signaling is contact dependent. So when the cells get into close proximity and the membrane-bound signaling molecule will interact with the receptor on the target cell, cell signaling can be triggered. A very famous example for juxtacrine signaling is the Notch delta pathway. So two adjacent cells where one has expressed the delta molecule, which is the membrane-bound ligand, and the other cell has expressed the Notch receptors. Notch delta signaling is involved in embryogenesis. It is very important for cell differentiation and can easily determine cell fate dependent on which one of these parts, the receptor or the ligand, is expressed.
Some cells are close and have direct contact. In the case of these two animal cells, they are gap junction close. Gap junctions in these animal cells, or plasmodesmata in plant cells, are connections between two close together cells that allow ions or other small molecules to pass. And by doing so, they don't have to pass across the cell membrane.
This is regarding cell signaling.